So, chain elongation. Probably should have gone back one more slide so that we looked at the chain initiation. But when we're looking at translation, so our final push from going from RNA into proteins, we're going to go through an initiation of that protein synthesis, the elongation of the chain, and then the termination of the chain. And remember, you've got all these little contributing factors along the way. Within our initiation, what do you remember from the initiation? So FMET in prokaryotes, the very first amino acid is FMET, so formal methionine. That formal group is there to pretty much shut off the reactivity of that methionine, initial methionine. So the nitrogen is then blocked off so it can't react with something else in solution. That's also going to act as a marker to say this was the beginning of the protein sequence. I can then cleave that off later on to get it to the proper nature. Yeah?
And it starts at P.
Yes. So within our ribosome, we have 3 binding sites for prokaryotes. We have E, P, and A. E stood for our exit. P stands for? Sure, why not? Exit, I've been referring to it as peptidal. It doesn't really matter as long as you pick up the P some way or another. And then A was the acyl, because that's our tRNA that's bonding our amino acid, and so that's the new one coming in. So our A is our entrance, P is where our peptide is being formed and then pushing out of the ribosome, and then E is our exit. Okay. In prokaryotes, we have these 3 distinct sites. In eukaryotes, it changes a little bit. We don't have a distinct exit site. We really just have the A and the peptidal. So as soon as you transfer the polypeptide from the E to the A site, the P to the A site, the empty tRNA just immediately gets kicked out, okay, in the translocation. It doesn't stall within the ribosome. How does the ribosome recognize the DNA, or RNA, sorry? The start codon is important because that's where our initiation is going to start. Our first amino acid gets incorporated. But that's not what our ribosome is actually looking for. That's such a small sequence, it's not going to be able to actually decide that that's where it's supposed to start. There's a recognition sequence that's known as the Shine-Dalgarno sequence in prokaryotes. Okay? So our ribosome can recognize that site, come together, bring in the FMET tRNA, officially FMET tRNA FMET, because one is saying that the amino acid is there, the other one is saying that's the type of tRNA, just so you know. And we've now got everything put together. We can bring in the next acyl tRNA to transfer the next amino acid on, and we can kick out the FMET. Okay? And that's part of our elongation process. What other things might contribute to the initiation? Would there be other things that are going to contribute to help bring these things together? Enhancers are looking at DNA. We're at RNA now. Kind of the right idea. Concentration is going to help. We're looking at some kind of factors that are going to facilitate initiation. And what are we going to refer to those as? Initiation factors. So you get IFs. Okay, you get a sequence of IFs, and I think they list them out. I don't even remember what they are. When we move up to elongation, we're now going to have a very similar process. We're now repeating this sequence through and through, except now we're going to have elongation factors, because now we're making the peptide longer. So, of course, we have to change the factors used. That's where we're getting our elongation factors. For prokaryotes, there are 3 primary elongation factors to worry about: our F2, our FP, and our F, for lack of wanting to spell all those out. The F2 is part of fidelity, so it's going to help ensure that our tRNA matches the site, and it's also going to help us it's going to make sure our tRNA matches the site. If it doesn't match the correct codon, what happens? it doesn't release the tRNA, and so the EF2 ends up taking the tRNA back out of the ribosome, and we don't get the amino acid adding on to our chain. If it does match, we go through a hydrolysis of GTP, which then allows for the separation of the F2. Is everybody okay with me saying F2? Yeah. Okay. I'm just kind of lazy, I guess. The separation of the F2 away from the tRNA, and our tRNA is now in the A site. We now need to actually get the amino acid on that new tRNA attached to the peptide chain. That's where we bring in more elongation factors. EFP is allegedly primarily responsible for that growing chain. Okay? So that's going to take the peptide that's at the P site and link that to the peptide at the A site. Okay? So it's probably part of that catalysis. At that point, we can then do a translocation. So what we'll end up doing is shifting the RNA through the ribosome, which means whatever was at the P site shifts to the exit site. Whatever was at our acyl site shifts to the peptide site. That word. Okay. We've now effectively reset. We're back at where we were officially at the beginning, and we can now do the next process. And the next tRNA being guided by F2 can then come in and help facilitate that peptide growth. Okay, so this cycle repeats again and again and again until until it stops. Okay, so there has to be something to recognize that stop. What are we looking for in the stop? We're looking, so hairpin sequence of use is all great for termination in transcription. We're at translation. How do we know to stop in translation? We're looking for the stop codon. So there's a sequence of three nucleotides that is going to have a particular attraction for a particular tRNA or another transcription factor that is going to prevent the amino acid from continuing out. So what we're looking at then is your termination. Okay? So what will happen with your termination is that you encounter one of your three stop codons, okay, which, like you, Stephanie, there are some U's in those. Not a lot, though. And our release factors. Okay? I don't know why I decided to call them release factors, not termination factors. But what we're looking at is release factors will come in, bind to that site, and now there's no amino acid to transfer across, and we end up shutting down the peptide synthesis. Okay? At that point, we'll again get a translocation that kicks the peptide chain out, everything falls apart. One of the things that's helping to catalyze the reaction is our ribosome has a phosphate on it. It will maintain that phosphate until it hits solution. Once in solution, we'll cleave that phosphate off. It's now back in its dead state, and it can go through and find another mRNA to go ahead and link up with and restart the whole process. Okay? Questions about that? Okay. So, initiation, IF. Elongation, EF. Termination, RF. So what we're looking at is trying to see roughly what happens within each of those stages. Most research has focused on the initiation and elongation. We haven't seen a lot on the termination, which is why we have a little bit less information on it. Okay? Questions? Because his car wouldn't start.
Well,
Hopefully Braden's mom or my mom. So, the components of our protein synthesis, you got a relatively nice table that goes through and lists everything out. We're going to have to start with our amino acid activation. So we're going to link our amino acids to our tRNAs. Our initiation, Fritos for breakfast, huh? Our initiation is going to be binding our ribosome to our RNA, and then bringing in the initiation, or finding the Shine-Delgarno sequence, bring in our initiation codon, our start codon our FMET, all that fun stuff. I'm just going to stay away from you guys with your bags. We can then move through to our chain elongation, what we end up needing for all of those, and then our chain termination and all of those individual pieces. Okay, so it just kind of lists all of the fun pieces. Officially, you are responsible for all of this, and in a future class, you are probably expected to be familiar with most of these terms. Their exact meanings and all that stuff is probably going to be more important then. What you should be picking up out of this is the general patterns and some of the major named sequences within these. Okay? In the prokaryotic system, our translation follows the transcription immediately. Okay? Why might that happen? So we don't want to worry about degradation, but in eukaryotes, you're going to have the same degradation issue. That's an interesting idea. Shorter lifespans could potentially contribute to it. Where is... What we're looking at is where these individual processes happen is probably one of our biggest contributors. In eukaryotes, we have a nucleus. So we're looking at replication and transcription all happening in the nucleus, and our translation happens outside the nucleus, where we have a higher concentration of our amino acids. When we look at prokaryotes, do we have a nucleus? No. So there's nothing to separate those individual components. Everything can happen in one giant big soup. Okay? Which means as soon as the mRNA comes off of our DNA from our transcription process, that mRNA can now immediately be picked up by a ribosome that's going to go through and start to process it. Okay? And start to spit out the proteins off of that. The other feature of that is that once that ribosome moves past the Shine-Dalgarno and past our initiation, so it's midway through the protein synthesis, what's now exposed? The Shine-Dalgarno and our initiation codon, or our start codon, which means another ribosome could potentially come in, link up, and start synthesizing that protein again. Okay? So with our prokaryotes, what we're going to end up seeing is, number one, because we don't have a nucleus, that translation will immediately follow transcription, okay? Happening virtually simultaneously. Okay? The other big difference is, or the other thing that we'll see, is that we can end up with something known as a polysome. Multiple ribosomes can translate the same strand of mRNA. Okay? So all we're saying by a polysome is we've got a bunch of ribosomes on the same strand of mRNA. And then prokaryotes, our big deal is our coupled translation, which we just addressed. We can have transcription happening at the same time our translation is happening. Can translation ever happen before transcription? No. Why not? Yeah. What is transcription reading? DNA and turning it into? RNA. What type of RNA? mRNA. Translation, what is it reading? Reading mRNA and turning it into proteins. Okay? So if we don't have the reading sequence for our translation, we can't actually start that. So the simultaneous is a bit overstated, but it's pretty close. Eukaryotic translation, so we might as well see more of this pop up. Of course, it's more complex. They were somewhat more intelligent with their naming system. When we're looking at the initiation, it's slightly different than our prokaryotes, but we will involve initiation factors, IFs. How do we separate those from our prokaryotes? Put an E for eukaryotes, or eukaryotic. Okay, so that's going to be the primary marker between those two as being different steps. Okay? They've gone through and found 13 initiation factors, which makes this a lot more complicated. You'll notice that we don't make any statements about those 13 because we don't really know that much about it to be able to be studying it right now. So it's definitely outside the scope of our class. How is it different from our prokaryotes? Okay, so one big thing is our aminoacyl tRNA, or sorry, our amino acid tRNA binds to the ribosome before the mRNA. And this is according to your textbook, by the way. I'm just trying to back up what the textbook says. In prokaryotes, the mRNA binds first, and then we can bring in our tRNA, okay, with the appropriate FMET on it. In eukaryotes, what we're saying is that the tRNA binds first to the ribosome, then we can bring in the mRNA to actually find the appropriate sequence. Okay, so it's a slightly different orientation or mechanism, if you will, in putting that together. Another big difference is we don't have FMET. Okay, so there's no formal methionine when we're looking at eukaryotes. It's coming in just as methionine. Okay, so we don't have to worry about that silly F floating out there. The other big difference, there's no Shine-Dalgarno sequence. What was the role of the Shine-Dalgarno sequence? Recognition of where to start the synthesis. Okay? Where to put in that first residue, that methionine residue. Well, if we don't have that recognition sequence, how can it actually discover where the start is? Okay? We get a slightly different mechanism. What will end up happening is this whole ribosomal unit comes in and links at the very beginning of our mRNA sequence. Which end? 5 prime. And what it's going to do is scan. It's going to go through and shift down the mRNA, and what's it going to scan for? Our start codon. Okay, but what's the difference between our start codon and methionine? And the methionine codon? We could go a little bit beyond very little difference. There is no difference between them. They are one and the same. So how does that ribosome know when to actually start the peptide synthesis?
As soon as the tRNA binds to the mRNA.
But the tRNA is just looking for the sequence AUG, which could be the start codon, and it could be a methionine that isn't actually in the sequence. Remember, our mRNA can have a leader sequence on it that doesn't get translated.
Is there like 50 S lines to it?
It has to be an AUG in the proper context. Okay? AUG through that sequence, it's not just wherever it shows up. Okay? It's when it shows up in conjunction with another particular sequence. Very similar to the idea of the Shine-Dalgarno. But because the mechanism of this scanning process is different than in prokaryotes, we can't call it the Shine-Dalgarno sequence, and instead it gets referred to as the Kozak sequence? Yeah, Kozak sequence. Okay. Why do we come up with a new name for it? A new person discovered it. Of course, we got to rubber stamp our names to everything. Okay. So in prokaryotes, we have the Shine-Dalgarno. The equivalent in eukaryotes is the Kozak sequence. Okay. Again, their functions are slightly different. The ribosome in prokaryotes seeks out the Shine-Dalgarno and then binds at that section of the mRNA. In eukaryotes, what happens? The ribosome binds at the tail end of the mRNA and just starts scanning through until it finds AUG, our start codon, with the Kozak sequence. What if there's no Kozak sequence? It keeps going until it finds an AUG with the Kozak sequence. Okay? It sounds kind of complicated, but in most cases, apparently, that AUG is one of the first sequences that you'll incorporate or you'll see at the beginning of your mRNA strand. Okay? So it's not like you'll skip 50 AUG start sequences to get to the proper alignment. Okay? It usually happens within the first 2 or 3 times that the AUG shows up. Kind of make sense? Okay. Once it's actually found the sequence where it's supposed to start, then the full ribosome can actually be built. Okay, and that's where we complete that ribosomal structure and we end up with the ADS. What's the ADS in reference to? The density of that final complex. How do we make an ADS complex? You bind a bunch of smaller complexes to it, which might be 50 and 30. Does it have to be 50 and 30? No, because remember it's density dependent and that structure can shift and change around and also ultimately change its densities. Okay? I'm pretty sure there is a question on Sapling that asks what pieces go into an initiation complex. Okay? This is where your textbook is going to be very useful because it will list all of the components within the initiation complex. So we can take a look at our initiation kind of prettied up, and here you can see all of the, or some of the initiation factors involved in this process. So what we end up going through and do is putting in our methionine. How do we know it's different from the prokaryotic version. If we take a look at our tRNA, it says met tRNA and not fmet. Okay, so in this case, just by looking at this figure, we should be able to immediately say it's eukaryotic because of the met. What else can we say that makes it eukaryotic? And don't tell me it says eukaryotic at the top. It says EIF. So we're looking at eukaryotic initiation factors in all of these processes. Okay. What's something else that you could make reference to? They don't show a really good job, I think, of that cap binding site, but you're right, we do see that. ADS? Oh, 80, I get it now. Yes, let's put that down. We could look at the total density of our initiation complex.
What is the prokaryotic?
I think it's 70S for the prokaryotic. I wouldn't stress too much about those exact numbers, just do realize they are different. Yeah. What else within this figure could help you decide that it's eukaryotic? Yeah, what? When did the mRNA get incorporated?
After the tRNA.
After the tRNA. So here's our mRNA. This class is swiftly falling apart. We'll notice that we get the tRNA being incorporated with our methionine first, and then at a later stage we see the mRNA come in. Okay? And then in each of those steps you'll notice they list off a lot of initiation factors. Okay? And it's not even as simple as just initiation factor 1, 2, 3, 4. What they've noticed for initiation factor 4 has a whole bunch of different components, so each of those then get listed with their own alphabetical sequence as well. Okay, yippee. Once we've got it initiated, we can then go through the chain elongation. You'll notice for the elongation and then termination that we have even less information, and I'm pretty sure even less images. Yeah, because we just move on after that. Why? There's not a lot of research on it. Okay. I don't think it's on the test either. There's not a whole lot of research focusing on it right now because we're trying to work piece by piece. Eukaryotes are much more complicated, which makes it more difficult to go through and process them. Okay? So we're focusing more on the initiation. Once we got the initiation solved, then we'll start pushing into the other factors. Within our elongations, we do have several elongation factors. They are named very similarly as we saw in prokaryotes with our EF, elongation factor, and then we also have the E out front, lowercase e, representing the eukaryotes. They went through and numbered these as opposed to using a lettered sequence, probably because of how they were discovered. Okay? In the prokaryotes, what they noticed was probably those elongation factors in response to particular temperature conditions or other potential conditions. So they came up with those names. EF2 was, TU stood for, not in this case, temperature unstable. TS, temperature stable. So it was probably part of how they isolated and found those elongation factors that they came up with those names. Since we probably found the prokaryotes first, when we shifted into eukaryotes, we just started listing out a numbering sequence because we identified them and weren't able to pick up the conditions as easily. EF1 is very similar to our EF2, sort of. It's similar to EF2 and EFTS. How could that be the case? There's two types of elongation factor type 1. There's part A and there's part B. Okay? So again, our nomenclature system gets a bit odd with that, but that's how they're referencing it. You've got those individual pieces coming through. Within the termination, the stop codons are the same as our prokaryotes, so that's kind of nice. Why might that be the case? Why are the stop codons the same?
Because of genetic codon.
Our genetic code is universal. Okay? It codes for everything across all forms of life. We would expect those stop codons to be the same based on our premise that we started with at the beginning. Okay?
Is that
What-
why it's so easy to have bacteria make like insulin and different things?
Yeah, because we know how to make it based on our codes, and we can incorporate those codes into them, and they will synthesize it the same way. There's only one release factor in eukaryotes, and it recognizes all 3 stop codons. Okay, so that's a nice little handy thing to have. And then there's one extra little factor. There are special tRNA molecules that will act as suppressors. So what they will do is ignore the stop codon and continue the translation process, okay, or facilitate the ignoring of the stop codon. Okay, so potentially if there was a mutation and we want to continue building that peptide chain out, okay, we might want that suppressor available so that it can fix that mistake. Once we've got the protein synthesized, okay, this slide has a whole lot of words on it, but really all we're saying in a very fancy method is what can we do to proteins after we have the primary sequence. So what can you do to a protein? Think simple first. You guys were already tested on this. Primary sequence becomes secondary, so we can get the structure folding on itself to generate our beta sheets and our alpha helices. What can then happen? You can get your tertiary structure, so your secondary structures then fold on top of each other. We'll also look at secondary interactions, like hydrophobic and hydrophilic interactions. We can look at metal ions binding to it. We can look at disulfide bridges forming between cysteine residues. Okay? So we've seen a lot of these modifications of proteins already. All they're going through and doing is now saying it's post-translational. It's the same thing that we looked at before. Okay? So pretty much if you can envision it, it can happen to proteins. Okay? So some of the things that we could list off, that F-met that we used to start, okay, that's going to change the chemistry of that protein or that amino acid at the beginning point. So we're going to want to get rid of that because we don't want that change happening in there. So one of the things that will happen in prokaryotes is we will cleave that amino acid off. Okay? Could we cleave off other amino acids? Yeah, as seen. Okay? The example that we talked about so long ago was chymotrypsin. Chymotrypsin gets synthesized in a non-active protein state. Okay, what happens when we hit the proper conditions? Sections of it get cleaved off and it folds again, and we end up with the active state of that protein. Okay, there's other forms. Our insulin. This I also find funny. We've got pre-proinsulin, we've got proinsulin and insulin. All of those are a peptide sequence with a slightly different sequence attached to the front of it. So we can cleave off those sections to push us to other things. Right? Our leader sequence can be removed. It's pretty much the same thing that we were just talking about. We could look at all sorts of fun places in the cell if we wanted to. Our endoplasmic reticulum, the Golgi apparatus, yada, yada, yada. Have fun with cell biology. We can incorporate secondary groups like our metal ions. Okay? Metal ions typically need something to coordinate to them. What charge would those things be? Negative, because our metals are all positive. So we could see cysteine residues helping out with that. We could potentially see histidine residues, okay, or acidic residues, depending on our pH, contributing to and chelating those metal ions. One of the big metal chelators that's responsible for how we transport oxygen in our bloodstream, heme functional groups. Those hemes can now be incorporated into the protein structure as well, along with that metal ion. What properties do we have of hemes? Yes, there's iron, fair enough. Okay. We could look at allosteric interactions, but what I was referring to is just the physical properties of the structure of a heme. Hydrophilic, hydrophobic. It is flat. It's quite hydrophobic. You've got a lot of just carbon hydrogens on the outside of that, so we can actually incorporate hydrophobic residues or hydrophobic components into our proteins. We might phosphorylate sections of the DNA. Why might we do that? or DNA, proteins. Why might we phosphorylate sections of proteins? It can do with activation, deactivation. What was our primary source of energy? ATP, and what part of the ATP structure is what's storing that energy? The phosphate bond. So if we're going to try and catalyze a reaction, we're going to look at the transfer of a phosphate group from particular sections. Well, what do proteins tend to do? They tend to be enzymes. What do enzymes do? Catalyze reactions. We need an energy source for that catalysis. So we can put phosphate groups on or off of a structure to help catalyze a reaction. They could also go through and activate individual sections within the protein, marking it for potential other processes. Right? We can change individual amino acids. Remember, we have 20 primary amino acids, but most of those amino acids can be converted into some other type of amino acid. The example that they're given here is proline to hydroxyproline. Right? So what happened to do that? We added an alcohol group somewhere within the structure of proline. Other covalent modifications, you can bring in carbohydrates, you can bring in other small molecules, you can bring in other proteins, you can do pretty much anything you want. Once we've got proteins, they're pretty much the workhorse. You can do anything you want to them. Okay? So if you can make it, it'll happen. Okay? I was trying to do Bob the Builder. Is it Bob the Builder?
Can you build it? Yes, we can.
Yeah, that's Bob the Builder, isn't it?
Yeah.
Yeah, see, I was trying to do that.
Yeah, if you build it,
What's that?
If you make it.
they'll call it. If you build it.
Yeah, I guess that's true. In any case, almost done, I promise. The last thing that we can look at, I do find it odd that we're bringing it in this section and not in back when we talked about proteins, but we can look at the destruction of the proteins after they've been made. There are several ways to go through and process or facilitate their destruction. The big one that they're mentioning is ubiquitin. So what ubiquitin will use or do is mark the protein, allowing it to then be recognized by other enzymes that will go through and now chop it up. Okay? So ubiquitin is just kind of the flag of here I am, kill me, kind of thing. So how that process actually works, we've got a sequence of enzymes that are going to go through and facilitate the transfer of that ubiquitin molecule to the appropriate protein sequence to be decomposed. And that protein sequence in this case is our substrate. Okay? Our proteosomes are where we're finding the highest concentration of our ubiquitin and are responsible for our protein degradation. We'll cleave off some ubiquitin pieces, which will get freed up as a single ubiquitin molecule, which will get picked up by enzyme number 1. Okay? The linkage between enzyme number 1 and that ubiquitin molecule is through a thio... Is it thioester? Is that what I wrote? It's definitely thio. Yeah, thioester. Why would it be a thioester?
Because you're using sulfur.
So sulfur, but then that sulfur has to be able to bind to the ubiquitin. So if it was just a sulfur-carbon bond, that bond isn't polar enough to then be reversible. Okay? One of the big things that we use in biochemistry for reversible reactions is the carbonyl functional group, because that carbon maintains a very good electrophilic reactive site. Okay? So we can use the sulfur to attack that carbonyl, create the thioester, which is now relatively easily reversible. Okay? Once we've got it onto enzyme 1, it will transfer the ubiquitin to enzyme 2. Okay? Again, we don't want it to be a permanent connection. So what we're going to do is do a trans-thioesterification, meaning we're just going to switch where our thioester bond is. So it'll be from enzyme 1 to enzyme 2. So the trans is just swapping the enzymes. Okay? At that point, enzyme 2 will bind up with enzyme 3. Enzyme 3 was there to actually hold the substrate. Okay? Get a hold of it and say, okay, this is actually ready to get destroyed. Enzyme 2 comes through, and now that complex is going to catalyze the transfer of the ubiquitin molecule to the substrate in a method that is not easily reversible. Okay? Which means we're going to transfer it to a nitrogen, and we're going to end up with an amide bond. Okay? So what does our substrate need to have to be able to pick up the ubiquitin? It needs to have an amine. It needs to have a nucleophilic nitrogen to be able to react with the ubiquitin carbonyl to actually facilitate that linkage. Once we have the amide bond, that's not an easy bond to break. Okay? What might be a good kind of sound logic behind why that could be the case? So what kind of bonds hold amino acids together? Yes, they're covalent. Push it a little bit further. They're amides. Okay. Are the proteins falling apart naturally on their own? Or do we have, say, a special catalyzed reaction that goes through and destroys them? Okay, we have a special catalyzed reaction. Why? The amide bond is not a particularly easy bond to break once it's formed. So what we're going to do is tag the ubiquitin molecule on there so it's not as easy to pop off. Okay? That way it's now permanently marked for degradation. Our proteasome can then finish up the rest of that, cleave the crap out of our protein and get us back to our amino acids. Why might we want to get back to the amino acids? So we can then reuse those parts in something else. So if we've got a protein that's no longer doing anything, we want to be able to recycle those parts. That's part of this process. Okay?
So is this all happening within like the lysosome?
You just brought in some cell biology that I have nothing about. I'm pretty sure we do get protein degradation in lysosomes as well. Proteosomes is another cell membrane, is it not?
Yes.
So you're probably seeing both things happening in both those cases. Okay? Or both, these things are happening in both of those places. To do this process, what did we need to have? The substrate needed to have what to pick up the ubiquitin molecule? It had to have a free amine. Okay, the substrate needed the amine group. The thioester was in all of our enzymes so that it could be easily transferred back and forth. Okay? We need that amine to be able to be marked by the ubiquitin. So we have to have that free amino group. Okay? Where might we find a free amino group? At the end of a protein sequence. So if that is free floating around, those are now susceptible to ubiquitin destruction. Okay? Where else might we see a free amine?
The other end.
The other end does not have a free amine. What's at the other end?
The carbonyl.
We've got officially the carboxylic acid of our amino acid. Remember, it's an amino and an acid.
So in the middle space?
Where in the middle?
I don't know. That's the other way.
The R groups. There are several R groups that have amino groups or free aminos on them. Any R group that has a nitrogen on it can now react. Okay? So what we've got listed up here, if we go through and take a look at the resistant ones, what do the resistant ones all have in their R groups? Resistant, no nitrogens. If we take a look at our susceptible ones, most of them have nitrogens. Some of them do not. What's happening with the some that do not? The some that do not have nitrogens turn out to be acidic. Okay? Why is that important? Well, there's another enzyme that goes through, or sorry, another tRNA and enzyme that will go through and recognize that acidic terminal group, and it will then go through and say, hey, we need to tag you with an arginine. Why tag with arginine? Arginine has a free amine in its R group. Okay? So that's going to help tag that protein for degradation through the ubiquitin cycle. Okay? How else could a cell go through, or we didn't do else. So how could a cell go through and prevent a particular protein from getting degraded in the ubiquitin pathway? Okay, if we have a protein that is really ridiculously important and I do not ever want it being decomposed, what could I potentially do? So I want to avoid amines. Okay, so we could go through and just say none of those susceptible amino acids. Okay, but even if we got rid of all of the susceptible amino acids, we still have an amine. Where's that amine? It's at the end of the amino acid, or yeah, the amino acid chain. So what could we do? We could put something on it that then covers that amino group. Okay? So we could come up with protection methods to go through and prevent that ubiquitin marking. Okay? Questions about that? Okay. So at that point, I'm pretty sure, yes, that would be the end.